Hello and welcome to another Aston Originals podcast here with me, Matt Derry. I'm a lecturer in chemistry here at Aston University. And today with us, we have a couple of people, Dr Goddard, who's a reader in biochemistry here at Aston. We've met Alan before, but I'll ask him to introduce himself one more time. And we also have, quite excitedly visiting us from France, is Dr. Mona Semsarilar. So welcome to Aston, and we'll hear from you very, very shortly.
Thank you.
So today, what we're coming to talk about is a specific project that we're actually here. That's why Mona is here at the moment. We're having our in-person kickoff meeting for an European Innovation Council Pathfinder project, which is supported by the European Commission and also by UKRI here in the UK. So we're having this meeting to kick off the project. It's a really exciting 3-year project that was awarded a total of 3 million euros. We've had a recent press release about this, and this is kind of the follow-up to that. Going to hear in a little bit more about our expertise and how everyone on the team are bringing those expertise together for this really challenging problem that we're trying to solve. So it also falls under the umbrella of the Aston Institute for Membrane Excellence, which we've also heard about recently. This is a new research institute that we're establishing here at Aston University, and this is another podcast in that series. So hopefully you'll continue to hear from us regularly about the exciting science that's going on. So I said we're here to talk about the project, and it involves researchers from across Europe who have had visiting us this week. And the research problem that we're trying to solve is to develop a novel bio-inspired membrane platform technology, a membrane technology to selectively transport certain molecules across them. So we can extract these molecules from water. And that's a really fundamentally interesting problem, a very challenging one as well. But we're really fortunate that we've got lots of expertise from across Europe. 2 of which I said are with me today. I'm also involved in the project, but I'm here as the podcast host. I'll contribute to the science on the little that I know occasionally. But first, let's start with Alan, as I said, reader in biochemistry here at Aston. So please just remind us who you are and what your research interests are.
Yep. So I'm Alan. I'm interested in biological membranes, so the things that surround all cells. I'm really interested in the membrane proteins and lipids that make the majority of these membranes, and especially how you can use those in real-world applications, so biotechnology, and in this case, in making these funky bio-inspired membranes where we take biology and try and get it to join in a functional way with polymer chemistry. And increasingly, I'm interested, worryingly slightly, in polymer chemistry. So I'm spending much more time with people like Matt and with Mona, who actually know about chemistry.
Yeah, so we'll make no apologies for getting you interested in polymers, because that's our thing, Mona, right?
Yes.
So yeah, Mona, please just introduce yourself and tell us a little bit about you and your research.
So Mona Semsarilar, I'm a researcher at European Institute of Membrane, which is part of University of Montpellier and CNRS in France, south of France. So I'm a polymer chemist by education. But since I've joined the European Institute of Membrane, I tend to do material that are useful for membrane applications.
Great. And as I said, it's really exciting to have you here. Obviously, we know each other personally as well, so it's great to have you involved in the project. Mona and I shared the same research lab for only a few months, actually, but we've got to know each other pretty well. So it's great to have you here.
Thank you.
So the project itself, the coordinator of the project, if you like, is a company from Denmark called Aquaporin. And they're called Aquaporin, inspired by the family of membrane proteins, which we heard about in the last podcast with Professor Rosalind Bill, who's also here at Aston. She's using them to study things like the onset of dementia. And
if you see on the screen, they're the yellow ones.
They are the yellow ones. That's exactly what I was going to say, Alan.
I stole your life.
Of course, Alan's expertise shining through there. So check that one out if you haven't done already. It's a natural one that goes hand in hand with this too, with this podcast. And Aquaporin are this amazing company who have this technology where they've embedded membrane proteins within a water filtration device, and it can selectively transport water across it. So only water molecules transport across it, which means you can very effectively purify wastewater streams or any other water stream. So it's a brilliant technology. It's out there across the world. They're using it in many different things. I've seen one of their latest wacky projects is taking one of their modules up to space to kind of clean up their wastewater up in space. So then I guess people can be out there for longer, and purify their own water out there means they can do longer missions. So that's pretty out there and exciting, but it's just one demonstrator of the technology. So I guess, Mona, if I come to you now, because people hear from me and Alan a little bit more often. So can you explain, like you mentioned things like CNRS and your Institute for Membrane Research. So what does that look like, obviously, in France, and what can you maybe say one current project that you're working on?
Sure. So CNRS is the French National Research Center. So it's a national-wide research center that employs researchers to do research in universities. And IEM, which stands for European Institute of Membrane, is an institute within the University of Montpellier, co-founded by university and CNRS.
Okay.
So I'm employed by CNRS, but I work in university.
Yeah, and you're the equivalent of a professor there, so director of research,
Yes,
right?
yes. So since last summer, I am a professor. Yes,
Great.
say that.
Yeah. And then in terms of one specific project, I know we discussed it yesterday, maybe the one with Alan's new favorite molecule, triangulamine.
Yes.
Yeah.
So we are making these crystalline based material called triangulamines that then we can modify their surface, we can modify their hydrophobicity, their hydrophilicity using chemistry, and use this modified triangulamines in order to make things like water channels or adsorption sites. And then we embed them in some sort of polymer network, like hydrogels or organogels, in order to do the purification of specific molecules or removal of a specific pollutants.
Perfect. So polymers come into it somewhere, Alan, don't worry. But Alan particularly likes this molecule because if you see it written down or drawn, it does resemble a triangle. And I guess, Mona, you stack these to make these water channels?
Sometimes we stack them, and sometimes we just disperse them in a network, in a polymer network. So
great. And I guess it kind of, this creating water channels kind of maps with what we're trying to do, Alan. So if you introduce that, and I guess I'll test you, introduce why polymers are so important.
Okay, so what we're trying to do is embed biological transporters like aquaporin proteins into a polymer membrane. And the real challenge there is that plastics and biology don't mix very well together. So you need something to bridge the gap. And really the kind of secret part of our project, which I'm now going to tell everybody on a podcast, so not that secret, is we're trying to make some special polymers that enable us to link biology and polymers together. And that's probably all I should really say at this point. But it's to allow us to bridge that kind of physical property gap between a relatively stiff polymer and a biological thing that has to move around a lot. And hopefully having some special polymers with aid in doing that.
Great. And I think what they call that in the trade is a teaser. So keep tuned for any work coming out from this research project. But yes, and I guess that's kind of where I come in, and colleagues also on the project like Professor Paul Topham, we currently share a chemistry lab here. We make those polymers that can have different functionality, that can interact with the biological membrane or any other chemical species that we want to incorporate. So polymers are the glue, let's call it. Yeah. You going with that?
Yeah, we'll
okay.
go with that, yeah.
The glue that sticks things together. So in terms of the project, I mentioned it's a Pathfinder project from the European Innovation Council with funding across Europe, and that coming to the UK is from UKRI, the Government Research Council here. It's called BIOMEM. That's the acronym for it, but specifically, and I might need to read this out just to get it right, is biomimetic selective extraction membrane. So BIOMEM. So keep a lookout for this. There will be a website very soon, and we'll link this with the podcast, and the podcast will appear on there. Look out for our social media channels, certainly through the Aston Institute for Membrane Excellence, and there'll be some specific ones for BIOMEM as well. So we're looking at biomimetic membranes. That's the thing here. So, Alan, why are they so important?
So many extraction processes now use a whole series of kind of physicochemical membranes to separate molecules from water, and lots of molecules have very similar properties. So if you want to select one against a panel of other ones, you might need a dozen purification steps, for example. That means you've not only got to make a dozen different filtration systems, it's very energy, cost and labor intensive. And we'd like to reduce that to a single step using the kind of inspiration of biology, where these cell membranes I was talking about earlier have proteins specialized to do these jobs. So an aquaporin, for example, moves just aquaporins. The pink one in the image up there might be something like a sodium channel, which might only move sodium, for example. and we can move a single molecular species in one step, so reducing that cost by maybe 50 to 75%. The advantages there are obviously you can clean up things from water, you can take whatever you want out of the water, you could desalinate water, and Aquaporin's membranes are very good at doing that already. But these new membranes would really broaden the scope of the type of molecules we can move. So at the moment, Aquaporin moves water, but you might want to work on, say, a biotechnology process where you've made a high-value chemical you want to extract from a complex mix. And at the moment, you might have to concentrate your solution up. You might have to do 6 or 7 filtration steps. We want a filter that does it in a single step using a biological transporter. Numerous advantages to that. There's obviously a clean water angle, which is really important. But from a biotechnology point of view, biotechnology is often kind of synonymous with sustainability and removing our reliance on petrochemicals. But most of the processes fall down, not because of the fundamental biology of can you make something, but the cost implication of can you make it at a cost when it's competitive. And maybe 50 to 75% of that cost is what we call downstream processing. So when you've made the thing, you need to purify it. And if you can do that in a single step in a platform technology, you'll make all these brilliant biotech processes more commercializable, reduce your reliance on petrochemicals, and to kind of maybe oversell what we can do, save the planet.
I mean, yeah, well, that's a promise. But in summary, I guess, in my understanding of this, so reducing all of these multi-step processes to a single step to get highly efficient, both in terms of the time, but also the energy, scalable processes to try and transport single molecular species. And this is a real challenge across the community, across multidisciplines, so chemist, biochemistry, material science. Yeah, we're excited about it. As I said, this is officially the teaser, so keep an eye out for things coming out of the project. And clearly not one lab can solve this, which is why we've got this consortium of people from across Europe. We've mentioned us here at Aston in Montpellier in France. We've also mentioned Aquaporin in Denmark, but we also have a few other project partners specifically. So Copenhagen University in Denmark, who will be doing things like molecular characterization and modeling, so some scattering on these systems. Also Tampere University in Finland, who are experts in wastewater treatment, so really seeing what the membranes that we develop can do. Also DSM Firmenich, who are a company, and the specific branch of the company that we're dealing with are a global science and innovation leader in nutrition, health and beauty. So it's really a team effort, at least 6 different institutions, yes. So I've kind of touched on it a little bit, but how is it that we can improve on existing membrane technologies, Alan?
So as I was saying, in an existing process, you've not really got that selectivity and specificity of a membrane. It might select for small things or positively charged things. And by doing that, you obviously eventually get to your output, but it's very energetically expensive. It's commercially big investments. But the beauty of our membranes, as well as being selective and specific, is that you can have this concentration effect where a lot of membranes now either need a high pressure to drive things through or can only act at what we call equilibrium, where you get the same concentration on both sides of the membrane. The beauty about these biological transporters, you can really concentrate up your product or the thing you're purifying. So let's say, as an example, if you wanted to recover lithium from polluted soil to reuse it in batteries, if you only recover lithium at the concentration it is in the soil, it's still relatively low. But if you concentrate it up, you can then get it into a workable, usable stock of lithium that you can use.
Okay, so, and I just thought about this. So if it's a terrible analogy, tell me. But like, if everyone has their box of chocolate at Christmas, right? The Quality Street celebrations, other brands are available. And everyone has their favorite, but everyone's favorite is often quite similar to somebody else's. So if you want to find your favorite out of this big box, it might take some effort to go through and do this. But if we had a membrane that only let your favorite through, that's kind of what we're doing, right? You have, let's just say above is wastewater with lots of different chemical species in, but we want one of them. We put a membrane through, and only that one that we want, let's say, I don't know, what's your favorite Quality Street?
Oh, I'm not a big chocolate eater, mate. You
Okay,
know,
just say the triangle one.
The green triangle
yeah,
one.
Yeah, because it's triangle, I mean.
Yeah, triangle.
Okay, let's say it only lets
triangles. I only like triangles.
Yeah.
Around, I know that.
Just 3 sides, that's fine. Alan wants his the green one, the triangle, to come through, and we can have, let's say, a membrane to do that. So yeah.
My analogy is Guess Who. Other games are available. When you start with, you've got to identify a person, which will be your molecular species, and you start if you say, are they male? And somebody says yes, and you flip a load of people down. And then have they got red hair? And you flip a load more people down. That takes a lot of steps to get to your eventual person. our membrane is basically being given that name of that person to start with, and then you know exactly who it is and how you're going to get them out of that big bunch of other people to start with.
Yeah, much more concise analogy. You can decide when you're viewing this which one you prefer, but I like chocolate.
And triangles.
and Alan loves triangles. So clearly this is a difficult challenge, otherwise everyone would have done it already. So Mona, what are the challenges of developing membranes that have biology in them, or biomimetic membranes?
So as Alan mentioned, to marry biology to chemistry, that's the challenge. So we have got polymers that are often petrol based, so they've got certain properties, and then we have got the biological material that have got certain properties. So we need to bring these two together and match them together. That's where the problem is, and that's where the glue comes. So we need the polymers and the functionality of those polymers in order to glue the two together and hold them together.
Alan, you're outnumbered 2-to-1 polymers.
There's 3 of us. We're a triangle. It's fine.
That's true. Yeah, we are. So I guess this brings on to me talking why polymers are great. But yeah, let's say for a simple purpose, you've got a bit of spaghetti. A cooked spaghetti is like a polymer chain, but if you cut that in half, but don't cut it in half of it is, let's say, hydrophilic, will like water. Half of it is hydrophobic, will hate water. But this hydrophobic character can interact with things like lipids, which are important species in biological terms. So if we can anchor these polymers to the lipids that surround proteins, but also have them to be dispersed or solubilized in water. And this happens now. We're just trying to develop new ways to do this, to then have it in a water filtration membrane.
I'm going to ask you a question, Matt. What's your clever chemistry tricks for making the polymers exactly how you want them to be?
So we can play with the ratios of the chemicals that we put in. So I find it quite simple. But I guess maybe that's because we do it every day. Yeah. But we just literally, like cooking, we change the recipe, the quantities of things that we add to get a different product out there. And we can tune. We've got a wide range of monomers. They're the small molecules that basically are stitched together to make a polymer chain. We can choose many different versions of that with different functionality that we want. Chemical functionality. We might want certain types of functional groups depending on what we want to transport across. Thank you for the question. Table's turned. I'll go back to asking the questions now. So we're talking about water filtration, water purification. We believe and we hope that these will have a wide-ranging application. So you mentioned a little bit about lithium. Lithium's clearly a scarce resource that we use a lot every day in all of our phones, right? And it's incredibly important. So what other kind of things, like, let's say, in the biotech field?
I think the nice thing is that because biological organisms, microbes, plants, encounter almost any molecule you can think of, there are generally transporters for them. So we were talking like yesterday at our kickoff meeting about PFAS, which I can't remember what they stand for. So I'm going to turn to the chemist in the room to tell me what PFAS stands for.
Perfluoroalkylated polymer chain.
And they are described as forever chemicals because the bonds between the carbon and the fluorine are so strong you can't really break them down. But there are biological transporters that will take these out of the environment and concentrate them up. And that's largely because things like plants. So plants are great for transporters because they can't move anywhere. So if you're under threat or a microbe is under threat, it will move away from the problem. Plants can't move in that way. So plants, when they encounter polluted soil, for example, develop these transporters to maybe get rid of the molecule from their cells, or maybe to take up nutrients. So without wanting to say we could move almost anything, there are biological transporters for most things that we would find interesting. So anything that interacts with people, there'll be a biological transporter for it.
Perfect. And this research project really kind of, well, it is a project that's just starting, and it coincides with us establishing the Aston Institute for Membrane Excellence, AIME, and it really does like embody the interdisciplinary nature of what we're trying to do in this research institute. We've got chemists through to biochemists, molecular biologists, all working together for not a common goal, but the common goal is broad. We basically want to develop new membrane technologies, both for this, but also things wider spread. And you'll hear about these kind of things in follow-up podcasts, I'm sure, like drug delivery or drug development, things like anything that's a membrane, which essentially is something that you can allow things to pass through. And in our case, exquisitely, selectively. So it's very much within the AIME remit, which is exciting. And I think we've done a great job of explaining the project as much as we can. Again, this is the third and final teaser, and reminder to keep a track of research that's coming out. All that's left for me to do is thank Mona very much, and it's really good to see you here at Aston.
It's exciting and happy to be part of the project.
Yeah, great. And Alan, thank you very much.
Thanks, Matt, and thank you, Matt.
Yeah. And thank you for listening. Keep a lookout on our YouTube stream, Aston Originals, and across social platforms. And we look forward to seeing you next time.